Hi everyone, I'm Jasmine and I will be giving an introduction on Chip-Seq. What is Chip-Seq? Chromatin immunoprecipitation sequencing is also known as Chip-Seq. It's used to identify genomic DNA binding sites for transcription factors and other proteins. DNA-bound protein is immunoprecipitated using specific antibodies. Immunoprecipitation is used to separate proteins that are bound to a specific antibody from the rest of the sample. Bound DNA is then co-precipitated, purified, and sequenced. Primarily used to determine how transcription factors and other chromatin-associated proteins influence phenotype-affecting mechanisms on a genome-wide scale. Combined with next-generation sequencing, we find the exact binding site to which the protein of interest is bound. Chip followed by next-generation sequencing, so Chip-Seq, of the immunoprecipitated DNA is a powerful tool for the investigation of protein-DNA interactions. To perform Chip-Seq, chromatin is isolated from cells or tissues with or without chemical cross-linking and fragmented. Antibodies recognize chromatin-associated proteins of interest are used to enrich the sample for specific chromatin fragments. The DNA is recovered, sequenced on various next-generation sequencing platforms, and aligned to a reference genome to determine specific protein binding loci. Chip-Seq studies have increased our knowledge of transcription factor biology, DNA methylation, and histone modification. How does Chip-Seq work? It extracts specific proteins together with any part of the genome they are bound to from the cell. So we use DNA sequence attached to proteins to infer the site across the genome that they interact with by identifying regions of the genome that are overrepresented in DNA in sequencing data. Chip-Seq identifies the binding sites of DNA-associated proteins and can be used to map global binding sites for a given protein. Chip typically starts with cross-linking of DNA-protein complexes. Samples are then fragmented and treated with exonuclease to trim unbound oligonucleotides. Protein-specific antibodies are used to immunoprecipitate the DNA-protein complex. The DNA is extracted, sequenced, giving high-resolution sequences of the protein binding sites. Sample preparation and sequencing. We have our nucleosome, which is composed of 8 histones plus DNA. We have our TF, transcription factor, as well as our chromatin. Histone modifications control how tightly the DNA is wound around the histones. So, like I mentioned, to perform Chip-Seq, chromatin is isolated from tissues or cells and fragmented. Antibodies recognizing chromatin-associated proteins of interest are used to enrich the sample of specific chromatin factors. Samples are then fragmented and treated with exonuclease to trim unbound oligonucleotides. Protein-specific antibodies are used to immunoprecipitate the DNA-protein complex. The DNA is extracted and sequenced, given high-resolution sequencing of the protein binding sites. After we sent out our samples for sequencing, we generate computational analysis of all the data we've sequenced through next-generation sequencing. Disadvantages and limitations to Chip-Seq. Chip-Seq itself can be relatively costly, from $2,000 to $3,000 for a preparation kit. The quality of the data relies on the quality of the antibody, so we need to account for differences in antibodies between vendors, lots, and studies, as well as GC-rich reagents, high chippable regions, and non-random fragmentations can introduce biases or background noises. The advantages of using Chip-Seq are the resolution at the single base pair level. It captures DNA targets of transcription factors or histone modifications across the entire genome. There's a low amount of chip DNA that is needed to generate our data, as well as reduction in noise compared to another method called Chip-Chip. Controls use: input DNA sample. This is a sample that has been cross-linked and sonicated but not immunoprecipitated. Another control is IgG controls, which reduce biases and background, represent a random non-specific population. RNA-seq versus Chip-Seq. In lecture, we discussed RNA-seq. for next-generation sequencing. RNA-seq data is used to identify and characterize quantitative differences in gene expression between biological samples from two or more groups. RNA-seq also utilizes mRNA, and then we need to convert that to cDNA to run our next-generation sequencing. On the other hand, Chip-Seq data is used to access the interactions of a protein of interest with DNA on a genome-wide level. Now for some data generated by Chip-Seq. In the heat map matrix, rows and columns represent each Chip-Seq sample. The samples are then ordered by transcription factors, and the color labels are separated by transcription factors. All cell classes with only a single Chip-Seq sample were excluded from visualization. Subsequently, transcription factors for which Chip-Seq samples were from a single cell type class were also excluded. So the heat map of transcription factor dependent binding K-mers, similarly to K-sim-Jacquard between the Chip-Seq samples. The colored labels of the rows and columns represent the transcription factors. Data visualization is an important step to intuitively assess the quality and to interpret the biological meaning of Chip-Seq data. It offers a quick overview of signal-to-noise ratio of the enrichment landscapes at genes of interest or of any potential bias in blacklist areas. Chip-Seq data visualization. Screenshots of human H1 Chip-Seq profile loaded onto the UCSC genome browser. Each mark has a distinct enrichment profile. In Figure 5A, the H4K9AC and H3K4ME enrichments are associated with active transcription and located around transcription start site regions, while H3K36ME3 is enriched along gene bodies. H3K4M1 represents here a more diffuse pattern. Repressive marks in B are usually associated with broader domains of heterochromatin or repressed gene expression. The last data set we will be looking at is Chip-Seq peaks obtained with this experiment show tight spatial association with transcription start sites. The histogram showing the distance relationship between the S-sulfouricus transcription factor B summit positions from MACS2 peak calling data and map transcription start sites. 450 out of the 1,035 mapped primary transcription start sites were associated with the transcription factor B peak summit in greater or less than 40 base pair distance. The skew toward positions upstream of the transcription start site represent as negative distance is due to closely spaced divergent promoters that result in overlapping transcription factor B peaks beyond the deconvolution capability of the MACS2 peak calling algorithm. There are many applications for Chip-Seq, as well as many other sequencing applications that can be used instead of Chip-Seq. Depending on what your experiment is looking at will dictate which method you will use for your experiment. And here are my references. Thank you.